starch, and I could say, why do certain organisms have to actually hydrolyze starch or make starch into glucose, and you would answer that. So it's more like, what do you see, a orange color, a yellow color, or whatever. It's more on like, so I know you read through the exercises, and you know at least the basics of what we're doing. I mean, there are like tons of biochemical tests, and they're just giving you an idea of what people use. So it's not that I think you're going to go out here and say, oh yeah, the blah 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 test is going to test for a gas production. It's more of giving you an idea of what the different tests are. But if you're more prepared, then it makes more sense than just, I don't know how many people did, but last time you could have gotten away by just saying stabbing agar or stabbing tubes and saying, well, I don't know what this is all going to do, but it was fun inoculating it for stabbing agar for five times. And this light now, this is responsible for 1 volt. Makes sense if you have 16. You need a whole line of switches here. Yeah. You get the artificial light, you get the window. So I don't like lecture after.
Me neither. I was so tempted to leave. I was gonna walk out of this.
The thing is.
Failure.
So close. I'm so close to a failure.
Where are you leaving?
Yes, man.
Yeah.
Oh,
This is
good
the
luck.
class that I'm really.
Good luck.
Alright.
Okay.
And the main point is, if you don't have math,
If you don't have math.
the main point is you don't have math. The main point is you only have to sit through 2 more after this. So it's pretty close, it's the end of the tunnel part. So this is 14, and it'll be a little bit shorter. But basically what we're going to cover today in Wednesday is the immune system. And just our way of, or the human immune system and what it does with the microbial, or the microbes that enter our body, what kind of defenses we have. Yes?
Are we not going to get to the biotech chapter then?
No, we will. I see. I have to somehow combine that with genetics. So I don't know if the genetics we can just cover real loose because it's so intense anyway, and I don't think it makes sense to. And then we'll just if we go through the main processes and then can talk about applications in biotech, that should probably work out. I have to do which one? I have to do at least one more.
Give us a heads up. When?
Getting out of hand. I'm glad this is over. But at some point, I'm gonna sit there and you guys are gonna give the exam. No answer. Well, what is it today? I don't even know. I haven't thought that far ahead. So you basically have 2 more days of lecture left before. And then I would say don't already ask me for a study guide because you have so much. I know.
Don't ask for study guide.
No, it's just because I can't correct this or I can't go through this and give you a study guide. No, just add the 3 study guides and then no. But what I would do is I thought I did this in this exam. You can tell me if that's not true, but maybe not so much for the chapter 8, but it's more general. I didn't ask a lot of questions about specific germicides or chemicals, or it's more what do they do, or if it's so. I think that's what I want to do, especially at the end, but not so. I mean, for those last 2 chapters, you're going to see a little bit more specific because they weren't asked questions for, but the rest is going to be more general. And I think I can write out basically if you want to start studying, you can go back to chapter 1 and say what do we really cover. 1 through 4, whatever.
Restudy those study guides.
Yeah, but then, I'll definitely give you more of like just big theme study guide where I say, what's really important in this, they say the second chapter there was a lot of, macromolecules. You almost won't go back there because I'm not going to ask you about the structure of starch in comparison to cellulose, but you've already gone through, the big carbohydrates, and so, some of the beginning work we did was to understand later chapters, so it wouldn't make sense to ask about those. You know what I mean? If I ask you again about the structure of or the bonds between, amino acids, I'd rather say, our amino acid or our proteins are what is the role of proteins in general in organisms, and you would say, well, they're mostly enzymes and have some other structural, but it's more general. But I'll let you know about those. So and I have to look at a final first anyway to see how I'm going to structure it, but it's probably a little bit like this or close to what we did. So just keep in mind that's important for this chapter. It's again more of keeping order in your head. So what's important is I'll give you an overview and talk about the specifics about these defenses, but you basically always have to remember that there are 3 lines of defense. So you have, and we're going to talk about the first 2, so you have one that basically are physical, chemical, and genetic barriers, and that's, if you think about first line of defense, then you can also think, well, where are the microbes, where's the microbiota, where are the portal of entry, and then think of what happens. We said maybe the oral cavity is one of the major ones, and so you have saliva. That's the first line of defense. Or you have other areas where you have a lot of microbial activity, and so you have just basic the skin, organs like that. So that's your first line, and that doesn't really. There's no real specificity to it. There's no. They're just. You'll always produce saliva, and it'll have some effects on the microbial flora that you encounter, but it's not specific. And then a more specific defense is the second line, and that's the innate immunity. So it doesn't have to be produced. You have it. It's not acquired. That's why I'm trying to make the difference here between the acquired one and the innate. So the acquired one is the one that you would expect to recognize a pathogen that you already encountered. And what do we do if we didn't encounter it, but we want the body to think it already saw that microorganism? So we vaccinate. So that's the third line. So we vaccinate with some kind of, for example, dead virus, so that it doesn't affect us, but that our immune system can produce cells that immediately recognize that virus if it actually shows up. So that's the third one, and we're going to talk about that Wednesday because it's more complicated. But today we're just going to look at the first line, the different physical, chemical, and genetic barriers, like the saliva or the skin as a physical one, and then you have the second line, and there you have those processes. You have inflammation. What does it mean if you have you have an inflammation? You have an inflamed area on your body. You have phagocytosis. So you have a way of actually you have cells that are able to ingest, for example, viruses or bacteria, and then you have other complement and interferon are basically or parts of your blood system that can also help fight off pathogens. And you can see over here, that's a pretty interesting picture of a neutrophils. So that would be one of those white blood cells that are attacking. And in orange you can see, like, I don't know what, I think it's anthrax, but it's a microorganism that's basically getting ingested. So that's what we're mainly looking for. How do these cells actually take in these organisms and get rid of them for us? And the most interesting, or if you think about it, just before we go into it, we talked about, I can't remember how many million microorganisms are in a milliliter. So if you think about like how, and then also in terms of evolution, how late we actually got here and how we have to deal with this constantly, it's amazing how our body actually makes sure that we stay clean. I mean, or that we stay healthy. It's amazing what kind of systems are in place and how complicated they are, but they keep us healthy. And so just always keep that in mind. So the 3 lines of defenses are basically what we're using. And I'm going to have a lot of repetitions just so you've heard it more often. So the natural defenses, innate defenses, are present at birth and not specific. So there's certain things that you see on, yes?
Clarify which chapters we're studying which day, so I can be prepared.
You mean, for this?
Today we're studying chapter 14.
So I'm not sure.
Wednesday we're going to study chapter 15.
Yeah, that would be the acquired immune system. That would
and then Monday we're going to do 9,
kind of like
9 or 10 or both.
I think because I know one person really wants to see the biotech. No, my approach will be to try to combine those, because we could go into all the details of the genetics, but maybe we can just cover the most important things and then show or talk about how they work and how biotechnology uses them. So the genetic part. Huh?
I can read it online.
No, that is. You can also just add a lecture right before the exam on Tuesday. No, I think it's still gonna work that way. I think that's more interesting too, but it's the tentative schedule. You know how that's always. But that's the schedule. So for the last one, you would just go through the slides and whatever. Like if you would study that for the exam, and then you would say, okay, that's all he covered. And I'm using the book again to see if what he said was right and whatever, like understand it. But you'd have to trust my slides more than the book and for that last one if we combine them.
Thank you.
So the innate natural defenses are there at birth, and they're not specific. So there's certain, for example, we've talked about those before too. There's certain for example, proteins on microorganisms that make your body realize these are not my own cells, and so they try to either reject or destroy, etc. So that's what the innate defense system does. And then the adaptive, we already talked about that too. It's specific and must be acquired. So that's specific against maybe the protein of the capsule of a microorganism or a virus. And so you can read here about the differences, but these I don't think of these non-specific first ones. I don't think of them first. I would always look at inflammation or phagocytosis. I would always look at white blood cells and think they are the ones who help me in terms of fighting off microorganisms. But you do have these, tiers, these substances that actually help you initially not to acquire any microorganisms. And then the second one, you have the white blood cells and certain mechanisms of your body like fever, inflammation that help you fight those that got inside of your body. And then the third is the ones that help you once they're inside your body and they're destroyed. If they come back, you already have an answer to them. So it's a little bit faster. And that's the last time I'll show this. I mean, it's going to be at the end of it too, but first line of defense, physical barriers, chemical barriers, and genetic barriers. Second line of defense, so you have inflammation, you have, it's almost like you have proteins, signaling proteins like interferons or proteins in your blood, complement, that are able to recognize microorganisms or foreign cells, and then phagocytosis is just one of the mechanisms that a lot of the white blood cells use, and we talk about those. And then the third is the one where this is what we're going to cover on Wednesday, and it's all based on B and T lymphocytes. So those are the B cells, and the T cells are the ones that are actually memorizing what was already encountered, and then they're able to produce a reaction. So those are the really specific ones. But we're only going to cover this part today. And this is a little bit more complicated, because to get to the point where you have a immune system, or you have cells that memorize what already was encountered, what structures, and then how they go from seeing that structure again and producing a response, is a little bit more complicated. So we'll have Wednesday to cover that. I mean, I think there's another slide about first and second and third line of defense. Overkill. Let's see. I think so. That's clear. We have those first barriers at the portal of entry usually, because that's where we encounter most of them. And then once they're in, the non-specific line defense, and then the third one we talk about that. So we'll start off with the first line of defense. And you could really think of, if you study that, you can just go back and say, well, where do we have a lot of interaction with microbes, and then you go from there and determine or keep a few of those chemicals or physical barriers in mind, and that should be making it easier to study them. Because it makes sense. We have a lot of bacteria. We encounter a lot in the oral cavities, so we'll have saliva. There's a lot in the respiratory tract. We talk about eyes, and that you're basically trying to keep microorganisms from the air out of your eyes, or what mechanisms are there to help you do that. And then in the digestive tract, always a big area for microorganisms. So skin is obviously one of the biggest ones, and what's important about the skin is that it has epithelial cells, or is composed of those, and they're kind of cemented together, and there's a substance, keratin, that makes it really hard for pathogens to enter. And then you also have sweat glands that are constantly, producing sweat, etc., to substances to actually flush out microorganisms. And then for the eyes, you have tear production, blinking, and then for the digestive tract, we already talked about the stomach acid, and there's a couple more. So it means in all these areas of interactions with microorganisms, you have substances and mechanisms like the blinking of your eye that constantly work against microbial invasion. And then if you look at areas, there, like in this case, the trachea, you have just the way, if you look at a microscopic view of the trachea, you can see, for example, how there's like these microvilli, and then you have the cilia. So there's a lot of, and especially because you're breathing in a lot of microorganisms or microbes. The physical structure of your respiratory tract up in this area prevents a lot of that to just go down into the more sterile areas in your lungs. And then what happens, whatever is collected here is actually through different mechanisms brought back up. So you have it, it comes back up here. Sometimes you cough, you sneeze, you can spit it out, but it's basically a transport mechanism that brings them all back up. It's called the ciliary escalator sometimes because it brings things back up. So there's a lot of mechanisms like sneezing, coughing, even vomiting is basically the body trying to get rid of a microorganism and toxins that are produced by them. And then what's also important is that you have a mucus coat. So you have, for example, in the onset of a cold, you have a running nose. It's your body producing liquid to actually constantly try to shed microorganisms. So that's part of response to nasal hair. So there's a lot of, probably eyelashes. There's a lot of different structures in your body that you wouldn't think of initially as supporting or facilitating the immune response or supporting your immune response, but they do. And like I said, it's probably easiest to remember it together with where the most microorganisms are encountered in your body, and then say, well, what happens in the oral cavity? How is there a control of microorganisms in there? How are they controlled in the intestines? Things like that. So chemical defenses that are not specific. So you have these sebaceous secretions on the skin. You have mucosa. So you have a film. You have in your saliva, in your tears, for example, you have lysozyme. It can hydrolyze the cell wall of bacteria. You have an acidic environment, and certain other components in sweat that make it hard, or that are germicidal. You have, in addition, or part of this creates this acidic pH on your skin. You have, for example, compounds, or you have acids in your stomach and in your intestines, including the bile, that are constantly, disinfecting or sterilizing certain areas. So you have that mainly done by acids, and then even semen contains antimicrobial chemicals. They found you have acidic pH. We talked about that for in terms of safety for the fetus. So you have a lot of different areas where microbes are controlled because you have, and especially in those areas, I guess, where you have a lot of microbial entrance. And then we talked about that, you also have a genetic defense. It goes back to, what if we bite a dog? And I looked that up a little bit, read a little bit more about it, and it seems like there's actually infections of, for example, monkey populations from humans, and they didn't get bitten by humans. So I think there's other ways. It's
like
just
what?
Huh?
Like
Well, probably in more of a zoo environment, but I can't remember what the disease was, but that would have been like almost something like influenza, where it makes sense that it's for a primate, but because the relationship or the closeness. But I think really what they usually call the zoonosis, what they usually call, or what you usually imagine is that we acquire diseases from animals, but they don't from us, and it's not really true. It's just probably the extent of how often it happens, and we're always more concerned about ourselves. That's an additional factor. So, because pathogens have a great specificity, there's usually some. They call that a genetic defense, but what it really means is the pathogen has a host that it's specific for, and so it wouldn't affect us. We could be handling animals with a certain disease and never ever get it, because the pathogen is specific for that for that organism or for that other host. And sometimes, and that includes what I said here. A lot of times the hosts are genetically immune to a disease of another host, so that would be an example of us not transferring any diseases to other organisms because those hosts are different and wouldn't be affected by it. And then there's even some differences where you have people with a certain genetic setup that makes them less susceptible to diseases like tuberculosis or fungal infections, etc. So there's that too, where you have just populations even that have less of a susceptibility than others. And
Like
it's just
sickle cell? Like a sickle cell?
Yeah, for example, with malaria, that you can't, you wouldn't, they wouldn't be. I think they're not sickle cell
Right.
carriers. Are not.
You can't get malaria if you have sickle cell.
Yeah, so they're not susceptible to malaria. So those would be examples for the genetic differences. What did they say about sickle cell anemia? That was on the radio the other day. It was, and I was like, oh yeah, I think they just said it's I don't know what the percentage in the population is, but they want to include it in the epidemiology. They want to try to make more countries aware of it, because it seems like a lot of parents don't know until later on that it's that's what's going on. And I think they said what I heard is that the medication for it is pretty cheap and really easy to come by. So and that's probably not the U.S. I don't know which countries are not treating it immediately, or I don't know what tests they would run here. So that's we've looked at some chemical, some genetic differences, some chemical options, and some physical barriers that we have, and that's all the first line of defense. You're not gonna, your body doesn't change its genetic makeup in response to a specific microorganism. So those are the first line of defense. They're innate. You don't have to. There's no process involved that first has to recognize the pathogen, etc. And then if you go to the second and third line of defense, that's what we really call immunology. And like I said, I don't usually look at the first line of defense as part of your immune system, but it definitely helps and can be considered that. So what's important really is, if you think about all these different areas, I mean, you're exposed to so many microorganisms all the time, and to different degrees of concentration in your body. You would imagine that it's easy to understand that your immune system is not a system like your detoxifying system, that's the liver, or your cardiovascular system, which is actually a little bit similar, but what in terms of how it's distributed throughout your body. So in the immune system, if you think about all these different portals of entry and exits and what's going on, you basically need to have an immune response in every single cell of your body, or close to every single cell of your body. So it has to be constantly surveilling your body. It has to be constantly checking your body where microbes came in, and always looking for basically non-self organisms, non-self cells. So it's constantly doing that, always looking around for something that came into the body that's not self, then recognizing that and then destructing it. So that's the process. And the main cells that are doing this work are the white blood cells, and we'll talk about those to a great extent in the next few slides. But what you can imagine is here, or what's shown here is if you have a self or a normal molecule, basically the white blood cell WBC is recognizing certain structures on that cell, but it would also recognize what they call pathogen, it has pathogen recognition receptors, and it recognizes certain proteins on pathogens. So that's important because whenever you have a non-self cell entering your body, there has to be a way for the white blood cells to recognize it and then to eliminate it or to destroy it. And so just before we go into it, a few definitions. The white blood cells is really what it comes all down to in terms of the cellular response, and the lymphocytes are part of those too. So the T cells and B cells we're going to talk about are white blood cells, but they're a different line, and we'll talk about those on Wednesday. But the rest of the white blood cells are the ones that are actually helping you in your immune response. Non-self and self, I already talked about it, makes sense, and that's the problem for, say, somebody who receives an organ and you have basically non-self cells, non-self tissue in you, and so the body is constantly identifying that foreign organ as non-self, and so you have to suppress the immune system. You have to suppress the work of the white blood cells, which then also is a problem because you're going to run into other microbes that are entering your body, and then you have a suppressed immune system and have to deal with that. And so what the microorganisms have in general, and it's a very general term, but they have pathogen-associated molecule patterns, or what it really means is they just there's certain molecules or certain proteins that you find on pathogens on the surface, and so those are shared by most microorganisms, and that's how white blood cells recognize those. So white blood cells are looking for specific patterns of what the surface of these cells look like, and if there are patterns that microorganisms show, and so those white blood cells can identify those with receptors. So white blood cell has a receptor for those. Yes.
So red blood cells is going to be cells, right?
Mm-hmm.
White blood cells. So white blood cells are called lymphocytes. So when they're eating everything, they're called beta.
Yeah. The thing is, and we get to that, the white blood cell population has a lot of different white blood cells. So there's some that are only looking for, some that are only more specialized in eukaryotes, the other one in pro, and then there's some that are phagocytosis or do phagocytosis. There's some that are eliminating them in a different way. So they might just signal to a macrophage, for example, which is one of those that uses phagocytosis. It might just signal to it to get to the site and then internalize and destroy the pathogen. But they are white blood cells, so they're always white blood cells. There's just different kinds, and I'll point out which ones are important. So two of those you probably haven't heard of, or at least one of them. But the immune system, if you think about like your whole body, and as I said, having white blood cells and a response to invasion available at all these different sites of entry, you have to have a pretty complex system, and it's got 4 major subdivisions. And so the bloodstream obviously carries white blood cells. You've probably seen that if you take your blood and you centrifuge it or you let it sit, it kind of the red blood cells sink to the bottom, and then you have a kind of a small layer in between, but then you basically have a whole layer of white blood cells. Also got a lot of water in there, but that would be one of the main ones. You've probably heard of the lymphatic system, your lymph system. So that's one that really, that's where a lot of times the microorganisms are presented to white blood cells, and then the white blood cells are doing their work in the lymphatic system. So here is where a lot of that happens, but those are connected, and I'll show you how. So the blood and the lymphatic system are connected. Everything that happens in the lymph system is always going back into the bloodstream. And then this is just it is major, but it's just, around every cell you have some fluid. So when you imagine microorganisms getting into your body and making it to basically to their final destination, which would be a cell, or in a virus case, we've seen how they infect or how they infiltrate. It's the cell that they're eventually ending up in. So you have extracellular fluid around each cell that is part of your immune system. I'll show you in a picture, but there are immune cells, white blood cells in this area. And then there's the reticuloendothelial system. That one you probably haven't heard of, and it's kind of like in the connective tissue between in certain organs that encounter more microorganisms. That's where a lot of the white blood cells are congregating, and so it's another area where you have a lot of white blood cells that can actually respond to invaders. So those four work together to keep your body healthy and to keep microorganisms in check. And we'll go through those. So this is there they are again, those four systems, and you have a direct, so this solid arrow shows you a direct connection between the lymph system and the blood system. So your blood system, you can imagine, you have these major arteries and veins, but you have the capillaries that go like on such a fine level that every single cell in your body is supplied with oxygen, and carbon dioxide is taken away. So the blood system already is really, it has to go literally to each single cell to be able to supply the cell with what it needs and to take all the waste out. And so the lymph system is a little bit like that, but it drains, so it goes, and I think there's another picture about that too, but it is spread out, but it doesn't have a major organ like the blood, like the heart, so it's not pumped. It's basically has other mechanisms to bring the lymph back into the blood system. And there's a picture of how that works. And then indirectly, you have, the blood is, like I said, it's getting really, all these little capillaries are getting really close to the cells, so the extracellular area is constantly in exchange with the blood, and it's also in exchange with the lymph system, or and also with the reticuloendothelial system, because this would be more of a tissue, a system in the tissue where cells are sitting. This is the extracellular area, so all of those are connected. And so exchange happens between the blood and the lymph, and between the lymph and the extracellular fluid, and then between the extracellular fluid and the reticuloendothelial system. And you can see that here. That's like trying to put them all together, but you would have tissue cells, and around the cells you have the extracellular fluid. Makes sense. It's extracellular cell, extracellular. You have capillaries that bring in all the gases you need and the nutrients you need, and then you have a lymph system. You have basically lymphatic capillaries here that are in exchange with that, and then you have cells here in between, like this one, that are white blood cells, and they are responsible for responding to invaders, and then they can there's an exchange between all these systems. So if you know those four systems, you can imagine they're all connected. It's not that one is separated from the other. So going back to that one more time, this shows where they're concentrated. So there's just areas where you would have more of this reticuloendothelial system. So it's a little bit difficult to understand. If you have epithelial cells, you basically have this base level of the cellular structure. So tissues are surrounded by that, and that's where this reticuloendothelial system is in place. So it kind of is around the organs, by the organs, and is usually denser where the organs that are exposed to a lot of microorganisms are. So if you have a, for example, the spleen is cleaning your blood out and usually mostly getting rid of old red or older red blood cells. It's also an area where you have a lot of that, or where that system is more prevalent. And in this reticuloendothelial system, you have a lot of those phagocytic cells. So you have mononuclear phagocytes or macrophages that are ready to attack, and we'll talk about those. Just keep in mind that that system is connected to, or is in the connective tissue, is by certain organs, or is surrounding certain organs, and is able to and is infiltrated with a lot of those macrophages that are able to ingest and destroy microorganisms. So what would be, I don't know why that's in here, should be a little bit earlier, but the first line of defense, what is not? So the first line was the very basic. Yeah, because they are the second. This is in your saliva, stomach, sneeze. So yeah. So it's more like a physical. Everybody okay with that answer? Yeah. So then, reticuloendothelial system. We've covered the blood is really important because it transports, just like the lymph system. It transports white blood cells all around. It has other important functions, such as bringing oxygen to each single cell, so they can do what? What can the cells do with the oxygen?
Just the breath, right?
Yeah, exactly. And then what do they produce that has to get away? Otherwise, it gets pretty toxic. Exactly. So that's where your blood is really, that's in the red blood cells. So going back to the Tour de France, if they do blood doping, a lot of times what they try to do is get more red blood cells into their body. So what they do is they train really hard. They take 2 liters of blood out, and then in the hotel, in between stages, they put the blood back into their body. And the problem is that they do it really, like they're athletes and they've done nothing but train, so they're not always the smartest. I mean, in terms of, it's really mean to say that, but what I mean is, like, they just look at the results. They don't really care so much, and there have been really bad cases of, I mean, people, the blood sugar, anything else, it's usually frozen. Somebody freezes it for them, and then they hang the blood bag somewhere so it can go back into their body, and then it's thawed out, and then sometimes it's not been done right, and they almost die basically from that. It happened a few times, but that goes away too far from. I like to talk about blood doping. The other thing, erythropoietin. Did you hear about that one? because it's basically a hormone that makes you produce more red blood cells too. So if you live like in an area where you need more because there's less oxygen, so if you grew up in Nepal, your body automatically has a way of constantly producing more red blood cells, so you have more oxygen available because there's not enough in the air, and so you basically just take that hormone instead of naturally produced, you take it in. They know how to produce it, and then your body thinks you need more red blood cells, and then you have more than the rest of the people around you, and that makes you deliver more oxygen to your muscle, and that can produce more ATPs, and then you're better, and then you're faster. And then there's also the oxygen tent. But anyway, back to the white blood cells, and that was all about red blood cells in your blood. The whole blood has basically the red, and then the plasma. And the difference here really is, a lot of times that's confused plasma and serum. The only difference is really that in the serum they've taken out the clot forming factors. So if your blood's able to clot a wound or whatever, you can take that out and then you have serum. But if you don't, it's plasma. You call it plasma. So it's just like a minor difference. And it has all kinds of. It's mainly water, but it has all kinds of proteins in it. Albumin is the most important one. Lots of proteins in it. It's got the gases, CO2, oxygen in it usually, and other waste products, but also other components your cell needs. And so that would be what that looks like. You know, if you spin it or if you let it sit for a while, you have the red blood cells sink to the bottom. You have the plasma on top. So the blood cell production is called either hemopoiesis or hematopoiesis. They use both of those words. And usually what happens is you have blood cells that are undifferentiated. So you have a main blood cell, a main stem cell, and that usually sits in the bone marrow. So in your bone marrow are stem cells that are able to produce different white blood cells, different types of white blood cells. But the actual differentiation and a lot of them, the maturation of those white blood cells into specific types happens in different organs. So it's not necessarily happening in the bone marrow, but that's where what they call it, the hematopoietic stem cells. So the production of blood cells and the stem cells that produce all the cells, which then differentiate in different types, happens in the bone marrow. And then for us, for the immune system, what's important are the leukocytes, and what they differentiate here are the granulocytes and the agranulocytes. And really, it's only differentiated that way because we stain them, and you see certain granula in some, and in others you don't.
So
but it's
two different types of leukocytes then?
yeah. Well, not yet, because there's more. But this would be the first differentiation. And actually, it's probably good because the next slide might be this one, and it's small. And if this is the slide, maybe you can go, whoever has their book, to page 441. 431, and look at that while we're talking about it, because it's too small here.
Yeah.
So this is just to differentiate between the different blood cells. So mainly, if you start here with the hematopoietic stem cell in the bone marrow, you can get basically a myeloid stem cell up here in a lymphoid. And we're not going to go into these. This is for Wednesday, but this would be your T and B cell. So we're just covering this side. but we're not concerned with the red blood cells so much because they use them for other purposes, or we use them for other purposes. And then there's also the platelet that the platelets that we use to close a wound, and so that one is not really part of what we're looking into either. So we're mainly just looking at the white blood cells that originate here, and
The yellow part of the
yeah, so the yellow part is all white blood cells, and basically you have got the granulocytes and the agranulocytes, so the ones that have that granule, and you can see at the bottom these pictures show you what they look like if you stain them. So it's mainly just a question there, how you stain them, but we're going to go through the different types. And so for the first part, or for the granulocytes, you see the neutrophils, there should be eosinophils, basophils, and they have different, those are different white blood cells, and they have different, first of all, if you do a blood smear, so you would take that blood and you produce a blood smear, and then you stain them, you would see a lot of neutrophils, for example. So those are phagocytes. They're some of the cells that destroy microorganisms by taking them in and digesting them, etc. And then the other ones, like I said, the eosinophils are, for example, those that are more geared towards eukaryotic cells, like they recognize fungal cells, and maybe parasitic cells, and then you've got the basophils, and then you've got what are really important are these ones, the monocytes, that then develop into macrophages and dendritic cells. So those are the ones that do a lot of the digestion of or destruction of microorganisms also. But you should, eventually you'll have to learn those, and I'm not sure yet. I think for this class, you can start with this class, but you really need to know like just the main components of the blood, and then just a general idea of what each single one does. And these ones we're gonna talk a lot more about, and then these other four probably a little bit less, but just to get an idea what their specific function is in the immune response and how they differ from each other. So far we've only talked about really what they look like when you stain them, and that's how they differentiate them in a blood smear. So if you, for example, would have a response to, or if they would look at your blood components in a test, they would probably look at the percentage of each of those, and sometimes that gives you indications of what kind of infections you're dealing with, because you have more of some blood cell type than of the other. So I think we're gonna cover those here anyway. So like I said, the neutrophils are pretty on a really they're the ones that you find the most, and that initial picture on that first slide showed one that was ingesting or destroying anthrax microorganisms. So that I don't think it's too important to know what they look like when they're stained, but it's important, they're the main phagocytes in your white blood cell, or among the white blood cells. You have about 55 to, well, they make up 55 to 90% of your white blood cells. The eosinophils are just 1 to 3%. They look a little bit different, and they're more geared towards eukaryotic pathogens. So fungi would fall into that category. Protozoans, that's where you would see more of a response from those. And then the basophils are really low, 0.5% of your white blood cells. and they're important because they release potent chemical mediators. So a lot of times, what you see with in an immune response, we'll look into inflammation. What you realize is that certain cells are already in place, or certain cells are there first, like the mast cells, for example, which are related to those, which are they're not moving. They're sitting in specific areas, and so if they respond first here in the connective tissue, so if a mast cell recognizes a microorganism, it would release chemical mediators or chemicals that would actually attract other leukocytes, so it would attract other white blood cells. And so that's more of what those two types do. They would attract neutrophils or macrophages to actually do the work. And then those were the granulocytes, and then the agranulocytes are the lymphocytes, the B and the T cells. And I already said that we're going to talk about those more on Wednesday. They're part of the acquired immune response. But what are important agranulocytes too are the monocytes and the macrophages. They're the largest ones. You see them in immune responses all the time. So you start off with the monocyte, and then the final differentiation. So they develop into macrophages, which are the biggest cells that are using phagocytosis to destroy microorganisms, and then dendritic cells are another part. So out of those monocytes, and you see that in the book too, I think, the monocytes divert into macrophages and dendritic cells throughout. So maturation is what creates those two, maturation of monocytes. So there's a lot of different cell types to look at, but you're always gonna come across those in other classes too. So it's good to know what those different cell types do, and they're all part of the white blood cell population. So you can go to a table like this one and just go through those and remember that those are the agranulocytes, and the granulocytes are up here, and they have different functions. And then just the platelet and the erythrocytes, we already talked about those. These lose their nucleus, the erythrocytes, and they travel basically through your body with facilitating and making gas exchange possible. They have a lot of hemoglobin. That's what's transporting your oxygen. And then the platelets are they're not really whole cells anymore. They're just elements that are formed from blood cells, and they're helped to produce these blood clots in cases of injury, etc. So maybe we can go through the lymphatic system too. So we did we covered the first two blood reticular endothelial system, and then the lymphatic system. And I don't think we're going to do much about we're not going to cover the extracellular fluid. I don't think it's even mentioned, because it's just there. It's just the liquid around that cells are bathing in. So to have a supportive route of return, or support for another system, just like your blood system or your cardiovascular system, you have the lymph system that provides another way of these cells to get back into the cardiovascular system. So if you look at this one, for example, you have your cardiovascular system with the arteries and veins and in the heart, and basically the lymph system is throughout your body, doesn't have a main pump, but has other ways of, it's got kind of like the same valves the cardiovascular system has, and so it's the site where a lot of the interaction between microorganisms and your white blood cells happens. So that's where a lot of it happens, and then it comes back into, or the fluids that are stored in there, everything comes back into your blood system. So it acts as a drain-off system for the inflammatory response. It acts as a place where where the responses can happen. We already covered that, so it does the surveillance, the recognition, and protection against foreign material, just as it happens in other areas. I think in the next, you can see it here. It's basically you have the lower parts and the higher parts of the body, and basically you have a lymph system that's connected, but it really in the end drains into a main venal part above the heart. And another really important, or one of the most important parts of the system are the lymph nodes. So that's where most of the rest is more for transportation. We'll go into that, but that's where the actual interaction between white blood cells and microorganisms happens.
So when your lymph nodes is swollen, that's because they're fighting an infection there?
Yeah, I mean usually it's where they sometimes they can't, or in general I would say that what happens is you have the interaction between them, and there's a lot of cells moving in to that specific area, and then it depends. Not all of them can be cleared right away, so there could be formation of nodes, there could be formation of concentration of all kinds of different white blood cells, in addition to the microorganism. So the lymph is what flows in the lymph system, and it's a plasma-like liquid. It's a lot of water again. And it's formed when blood components move out of the blood vessels into extracellular spaces. So it doesn't contain, it has this sort of like the same consistency and content as blood, but it doesn't have the red blood cells in it. So there's water, proteins, dissolved salts. We find all that in the blood, and then it transports the white blood cells, a lot of the infectious agents, and that's where they basically come together, where the interaction happens, and also cellular debris, just other, especially from, for example, microorganisms that have been destroyed. So it permeates all parts of the body. You have to have it just like the capillaries and the blood the gas exchange from blood. You have to have it pretty much in all regions of the body, except for the central nervous system, in the bones, placenta, and thymus. So those are the exceptions where you don't find it. What I said before, so since there is no actual pump for that system, you have they're easily permeated walls, but the contraction of skeletal muscle around it can actually sort of pump it up to this area up here. That's the basically the ducts that collect all the lymph, and then it flows back into the bloodstream. So all this liquid that or whatever comes from the blood and produces and whatever liquid that's produced by leaving the bloodstream and then being picked up by the lymph system and then brought into contact with white blood cells, is then transported back through all these different ducts, through the nodes, back into the blood system. So there's a constant exchange between the lymph system and the cardiovascular system. And then if you look at it, that might be see what's next. So if you look at the system, the lymph system, that might make the most sense here is you initially have these, and remember that round picture that showed the interaction between the extracellular fluids and the blood and the lymph capillaries. So you have these capillaries, just like blood capillaries, all over the place in your body, and they basically pick up extracellular fluid that came from the blood. So it's kind of like an exchange area, but they also pick up foreign materials that are in this area. So here's where it all starts. That's where they diffuse in, and then it all goes into bigger ducts that kind of concentrate all the lymph together into and transport it to lymph nodes. And this is here, for example, a good or a section of a lymph node that shows you what actually where these ducts go. So you would pick up liquid lymph and also foreign organisms, foreign material up here, and then it flows through these ducts to the actual lymph node. And then here, the foreign material is filtered out, recognized, and then it's processed by lymphocytes, macrophages, and dendritic cells. So remember, those were the main cells that basically take up microorganisms and destroy them. Those are the three main ones. And they process, they act in the lymph node. And then from there, slowly you'll have the lymph that's cleared, and then all the debris basically slowly dripping into these ducts further down. And then from these bigger ducts where all the lymph that was already basically treated in these nodes, they go back to, they flow, or they make the flow from lymph back into the veins possible. And then the main organs, you have to differentiate, or you can differentiate between where the where a lot of the white blood cells are maturing, where they're able to differentiate these different types of white blood cells, and that's happening usually in the area where it's produced. So the bone marrow is where all the blood cells come from, remember that. And then the thymus is, and don't confuse it with the thyroid. Thymus is somewhere around here, like the upper part of the sternum is in the middle, is where you would find it. And the thymus is where a lot of the maturation and differentiation happens. And then the secondary organs and tissues is basically where do these cells, where do you find those cells, where are they circulating, and where are a lot of them. And so that's usually, like I said earlier, the spleen, where a lot of the cleaning happens. The lymph nodes, that's where you need them. So the lymph nodes where you destroy the foreign cells, and then also in other areas where, like the mucous membranes, where you encounter a lot of microorganisms. And so what they call them the skin-associated lymphoid tissue. So that's where you would find a lot of the secondary products. So you find a lot of white blood cells that you need, and so they call that SALT if you take the abbreviation for skin-associated, and then gastrointestinal associated tissues are GALT, and then MALT for those that are found at the mucous or in the mucosal membranes. So just for the lymph system, just know how the lymph is produced, how it's the site of interaction between microorganisms and white blood cells, and then that the thymus is one of the main areas where those blood cells are differentiated and mature, and then also that there's areas where you have a lot of those cells congregating, and that's in these areas where you have a lot of microbial contact with the human body, like in the skin, the gut, the mucus. So the thymus has a high rate of growth and activity, so that's the organ that's important for the lymph system, and it's really important in the beginning, and then it begins to slow down its production. It's also the site of T-cell maturation, but we'll talk about that on Wednesday. And then the lymph nodes, we already talked about those. You've seen how there's a certain number of them along the lymphatic channels, and then the spleen usually filters and removes worn-out red blood cells and pathogens, but also has importance for because there's a lot of those white blood cells situated by the spleen. And then you have other areas where you have a lot of lymphocytes in area that encounter a lot of microorganisms. So the gut associated cells, for example, and then other areas. So it makes sense that in areas where you have a lot of contact with microorganisms, that you have cells that are part of the lymph system, or the lymph system to clear those up and to help in the defense. Yeah, that should be there because we didn't do the T cells yet. I mean, you don't need to. And then we'll start here on Wednesday, and then read a little bit about the lab tomorrow. I think we can go through the exam if you want to a little bit, because lab's not going to take 3 hours. And we have to do a little bit more for the incubations or the tubes we did last Thursday, but I think we have time for other things.